Hi, good morning everyone. I hope you're all doing well. So, we're gonna start with the further lecture. Only one student is there, but we will still start. Um, yeah, maybe you all will join soon as. So, continue with the fast performance liquid chromatography. That is it's advanced version of our HPLC, in which it's also a type of liquid chromatography where the solvent velocity is controlled by the pumps. The pump controls the constant flow rate of the solvents, and solvents are assessed through tubing from an outside reservoir. And the flow rate of the solvent is set through the computer inputs and controlled by the pumps. Further, in the gel electrophoresis, for protein separation, the most popular gel is polyacrylamide gel, which forms a three-dimensional molecular sieve. So, under denaturing condition, the gel can dissolve proteins of the size from 10 to over 250 kilodalton. That's even more advanced version of it. So proteins, nucleic acids, they could migrate over this electric field, and we can get to know what the various kinds of proteins, DNA, RNA, over the gel. And the most popular method that we use in the gel electrophoresis is SDS page. That is your sodium dodecyl sulfate polyacrylamide gel in this, basically, we are trying to separate large protein molecules, and the key feature is that the SDS tend to coat over the protein and then give it a negative charge. After giving it a negative charge, they run over the gel from negative to positive electrode, and then they're separated according to the size. So it's a free flow electrophoresis, and this is the simplest in the concept so far, but most difficult to achieve as it requires expensive equipments. And beyond that, we need also low ionic strength, which is passed through the two large glass plates separated by a narrow gap, which is applied across the width of the plates perpendicular to the buffer flow. So protein mixture is injected in at the top of the plates begins to flow down to the buffer flow. So proteins with that carry a net negative charge will also migrate towards positive electrodes, while positively charged proteins go to the opposite direction. So buffer flow is adjusted so that protein never reaches the electrodes before it arrives at the bottom of the plates, where different fractions are collected in small tubes. So at the bottom of the plate, a beam of UV light at 280 nanometers sweep across the absorption due to protein is absorbed. So gel electrophoresis. So we are having these negative and positive electrodes in which we have mixture of molecules, right? So we have small molecules, middle-sized molecules, big molecules. The small molecules, they go to the bottom, and the green ones in the middle, and the yellow one at the top. Yeah. So acrylamide, methylene, bisacrylamide. So they are the two ones that we tend to add. Polymerizing acrylamide and bisacrylamide together forms a network of pores, the size which defined by the initial acrylamide and bisacrylamide concentration. So you can see here's acrylamide, there's methylene, bisacrylamide. They tend to come together, and then afterwards they make this chain of our chain of a network structure. So which will let your protein separated from within this gel. and SDS is coded. So this is the SDS structure, sodium dodecyl sulfate, which tends to cross over your protein, and then this mixture is placed over the gel, and this is the direction of migration of your gel, of your proteins, and then according to the size we are letting them separated. So here's the SDS page. The key feature is that SDS coated proteins have the same electrophoretic mobility, thus molecular sieving affect the major parameter that cause a separation of proteins. So at the end results, that's what we see. Here is the molecular markers, and in this middle we can see increasing, uh, yeah, like the one protein is being increased in this amount. We can save them over the X-ray films, these data, for long keeping data and limiting them. This is different types of some results with different reagents. Yeah, so after we've gone through the basics of gel electrophoresis, now comes the isoelectric focusing gel electrophoresis. This is the first part in the 2D gel electrophoresis, two-dimensional gel electrophoresis. So before you do a two-dimensional gel electrophoresis, we have to do this isoelectric focusing gel, in which we separate the proteins according to their pH. So there is a high pH, there is a low pH on both sides of a strip, positive and negative, and then you can see these blue and pink and yellow particles. So they are having these positive negative charges. So they will find their isoelectric point where they are neutral in their pH. So and they will be stagnant at there once we run this over the this strip. So we can see a better results in that. We can see in the SDS page and in the isoelectric focusing gel. Here it's a modified EL5A protein is being highly coming up. So how we make the protein precipitation? To prepare your protein for the you know for the 2D gel electrophoresis. So for protein precipitation, sorry, it will not have the voice. Just go through these subtitles at the bottom. Okay. Now, once your proteins are precipitated and they are good to use for, we do the two-dimensional electrophoresis. So we are having this protein mixture of pH 10, pH 4 and pH 10, and we did this isoelectric focusing. Yeah. Then separated according to first dimension. Then apply first gel on the top of second gel. So we took this strip and put over this long gel, and then run for the SDS electrophoresis for the separation in the second dimension now by the size. Initially, we separated the molecules according to their different pH. Now it's 10 to time to separate them according to their charge, according to their size. So this is the results that we tend to see in the 2D electrophoresis. On the top, here results was because of isoelectric focusing, and the bottom it tends to separate because of the SDS page. Yeah. So here we can see various dots, like these dots belongs to various different proteins within the one human cell. Whichever the samples that you have taken, whether liver, human or heart, any sample or from mouse, so you can see all the proteins that are present at once within this. So this is actually a proteome of that, right? So let's now check very nice one professor from the BioRed company. So he will represent now how we can manage this 2D gel electrophoresis in the lab step by step from isoelectric focusing to the SDS page, so that you are able to, you are good in this technique, because this is highly like recommended technique being used in the labs. So I will start now the virtual lab of the same. So this will be a long 17 minutes video, so be patient and enjoy the video. So I will remove my mic from it now because of the voice of the.
Hi, I'm Steve Freeby. I am a staff scientist at Biorad Labs. In this video, I am going to demonstrate how to run good reproducible 2D gels. The video is going to be broken into 3 segments. The first segment will be how to apply, sample, and rehydrate the IPG strip. The second segment will be on how to equilibrate the IPG strips in preparation for running on the second dimension gel. And the third segment will be staining of the second dimension gel. In this video, I hope that you get tips and techniques and tricks that will help you run good quality 2-dimension gels. After sample preparation, the first step to 2D is to rehydrate the IPG strip. And I'm going to demonstrate this process using Biorad's 2D Starter Kit. The 2D Starter Kit is a great tool for learning 2D gel electrophoresis. It contains all the reagents and an E. coli sample for running a 2D gel. The first step is to prepare the sample and the reagents according to the instructions in the manual. The components of the rehydration buffer are urea, CHAPS, DTT, ampholyte, and bromophenol blue. I will be loading 200 micrograms of E. coli sample that has been reconstituted in rehydration buffer onto an 11 centimeter pH 4 to 7 IPG strip. To rehydrate the strip, I will load 200 microliters of sample into the focusing tray in each lane that I'll be running a strip. I am using a focusing tray that is specific for 11 centimeter strips. Trays are available for all IPG strip lengths. Tilt the tray toward you to allow for even distribution of the strip. An easy way to do this is to take the focusing tray, put it on the edge of the lid to the focusing tray, and you have the tray at an angle towards you. Dispense sample along one edge of the lane of the tray. And you want to dispense it in a nice even bead from one electrode across to the other. One even bead. I have shown you this with the focusing tray, but this can also be done with the rehydration tray. Now we are ready to place the IPG strip into the focusing tray. An IPG strip has two sides. One is a plastic protective backing and the other is a dried gel. You begin by removing the plastic protective backing with forceps, and you can tell where that plastic protective backing is by a slight indent on one side of the plastic pieces. The side that has indented is your protective backing. You pull that rapidly across, and now you have gel side up. Take your forceps, hold onto the strip on the negative side. You'll see the other side has a positive side. The negative side is opposite of that. And you can always tell when the IPG strip is facing down, you'll be able to read the numbers of the pH range of the IPG strip in the correct orientation. Now the gel will be loaded onto the sample bead, gel side down. If you are running multiple samples, I recommend labeling your strip before this step. Make sure to label the side of the gel and not the protective backing. To load the IPG strip into the focusing tray, place the positive end of the IPG strip into the positive electrode of the focusing tray and slowly drop your IPG strip into the lane of the focusing tray. Being very careful not to trap any air bubbles. If there are any air bubbles trapped, you can pick up the end and gently reposition the IPG strip until the air bubbles are expelled. If you don't remove air bubbles, there will be areas in the IPG strip that do not rehydrate properly, and that will cause serious anomalies in your first dimension isoelectric focusing run. The initial step of the first dimension separation is to rehydrate the strip. I will do passive rehydration in the focusing tray. Other forms of rehydration are active, which may be used for high molecular weight proteins, and cup loading, which is ideal for the separation of proteins that will be separated at extreme basic and acidic pH. Place the focusing tray into the protein IEF cell, making sure to align the positive electrode of the focusing tray with the positive electrode of the protein IEF cell, and add enough mineral oil to cover the strip in each lane. The purpose of the mineral oil is to prevent the strips from drying out during the course of the rehydration and the run. Now I am going to program the protein IEF cell. Instructions for programming are provided in the protein IEF cell manual. Typical rehydration times are 12 hours and focusing time ranges from 6 to 8 hours. At the completion of focusing, if you do not have time to run the second dimension gel, first remove the strips from the focusing tray, blot off the excess mineral oil, and place the strips into a rehydration tray at minus 80 degrees C. The steps following rehydration and focusing of the IPG strip are equilibration of the IPG strip in preparation for the second dimension gel, which I will now demonstrate. The equilibration process is a two-step procedure. Equilibration buffer number 1 contains DTT for reduction of proteins in the strip. Equilibration buffer number 2 contains iodoacetamide for alkylation of proteins in the strip. Each solution contains SDS for denaturation of proteins in the IPG strip to ensure optimal performance in the second dimension. Prior to equilibration, I will remove the IPG strip from the focusing tray and blot the excess oil off of the strip. First, pick up one end of the IPG strip, turn it over so the gel side is up, and place the IPG strip gel side up on a dry piece of filter paper. Wet another piece of filter paper and place the wet filter paper onto the IPG strip and gently blot the excess mineral oil off the strip. Be careful not to press too hard to damage the IPG strip gel. The next step is to equilibrate the strips. I prepared buffers from the Ready Prep 2D starter kit as instructed in the manual. Remove the wet filter paper from the top of the IPG strip. Place the IPG strip into a lane gel side up of the rehydration tray. Add 2 milliliters of equilibration buffer number 1 to each lane of the rehydration tray that you have a strip. Place the rehydration tray onto a shaker rocker table for 15 minutes. Note that the rehydration tray has an angled end and a straight end. Whenever you're pouring out liquids, use the straight end so that it does not slide out of the tray. Now add 2 milliliters of buffer number 2. Shake for 15 minutes and pour out. At this point, the strips are ready to run on the second dimension gel. I am going to use Biorad's Criterion precast gels, which accommodate the 11 centimeter IPG strips that we ran earlier. Prepare the gel by removing the sealing tape from the bottom of the gel and slide out the comb from the top. Rinse the wells with good quality DI water. Tap the water out into a beaker and then place the gel sideways and remove with a piece of filter paper the excess water from the wells. Be careful not to touch the bottom of the gel as it can damage the surface of the second dimension gel. We usually do this because unpolymerized acrylamide can cause anomalies in your second dimension gel if it's not removed. To remove the strip from the tray, place the forceps on the tray and gently push on the plastic backing of the gel to push the strip towards the angled edge. Lift up the strip from the right tab. I am going to rinse the strip off in running buffer that I have previously prepared. Dip 2 or 3 times. This will remove equilibration buffer number 1 and 2 that may still be on the surface of the IPG strip. Place the IPG strip gel side up. Place the plastic backing against the back of the cassette. Position the strip centered in the middle of the well with your forceps and gently push on the strip until it touches the gel. And you can tell when it's touching because you'll feel a little bit of springiness, a little bit of resistance. Don't push too hard. Now I will insert a Precision Plus standard plug, which is a broad range standard that has been embedded in agarose, into the reference well on the right side of the gel. Break off one plug, hold onto the handle and break off the spatula, and using the spatula, push out the agarose plug. For Criterion gels, I usually cut the plug into 4 slices to match the size of the well. In this case, I need 1. With forceps, take one of the slices, place on the back of the gel cassette over the reference well, and gently push the plug into the reference well. At this point, we want to seal the IPG strip and standard gel plug into the well of the gel. I will use Protein Plus Overlay Agarose. Heat in the microwave oven until it boils, and when the temperature of the agarose is no longer hot to the touch, take the hot agarose and fill up the well. Do this in one rapid action, side to side. Working quickly, use your forceps to push the IPG strip against the gel to push out any air bubbles. Agarose is going to solidify fairly fast. You can see the agarose filling in the place of the bubbles as they get pushed to the top. Small air bubbles are not an issue. Try to get some of your larger ones, and you should not have any problems for your second dimension gel. Now that the agarose is solidified, I'm ready to run the gel. In this case, I'm going to run the gel in the Criterion gel box, which holds 2 gels. In most cases, you're going to run multiple gels and you want to use a Criterion dodecacell. Remove the gel cassette and place it into the Criterion cell. Load the upper buffer chamber with running buffer. Place the lid onto the Criterion cell. Insert the leads into your power pack power supply. Program for 200 volts for 1 hour. Constant voltage. And start. The second dimension gel has now completed running. You know it is finished when the die front has reached the bottom of the gel. We're now going to remove the gel from the Criterion cassette and prepare the gel for staining. Remove the gel from the gel box, pour out the upper buffer, and open the gel by using forceps or a similar tool by pushing the forceps against the plastic cassette towards you at the top and work your way down. So fold open like so. After I open the gel cassette, I use forceps to remove the IPG strip from the surface of the gel. Starting at the bottom of the gel, gently pull the gel away from the surface of the cassette. The easiest way to do this, turn the gel upside down over the surface of the staining solution, remove a corner, and remove a corner until the gel falls into the solution. Biorad offers many different staining choices for 2D gels, ranging from the visual stains, the traditional Kamasis and silver stains, to the fluorescent stains, flamingo and cypro. In this case, I am staining this gel with BioSafe Kamasi, and the first step requires 3 10-minute rinses in water to remove SDS from the gel. We are now going to stain the gel using Biorad's BioSafe Kamasi stain. Following the 3 10-minute water washes, we're going to transfer the gel to a tray of BioSafe Kamasi, and we are going to stain the gel for 45 minutes to 1 hour. Following staining, we are going to transfer the gel from BioSafe Kamasi back into water, and we are going to destain until you get desired background. At that point, the gel will be ready for imaging. Here you can see that I have generated a gel with a clear background and well-resolved spots. I have now completed the entire process for running a 2D gel. I hope that the techniques and tips will help you when you run your next 2D gel. As you have seen, Clean Sample and the Biorad 2D Starter Kit will help you generate good quality, reproducible 2D gels. For additional information and techniques and tips on sample preparation and 2D gel electrophoresis, you can visit our Expression Proteomics website at www.expressionproteomics.com.
So that was the detailed version of um, proteomics, 2D gel electrophoresis. I hope it went normal, everything went well. Now I want to take you to the next part of this lecture. So these are the summary results from the various types of chromatography that we saw, from homogenate, from slot fractionation, ion exchange chromatography, molecular exclusion chromatography, affinity chromatography. But from the affinity chromatography, we see a single band which is highly accepted. So the more the band you see, the more clearly your results are. So some summary results of the protein purification. From homogenization, total protein that you get is 1500 milligram, 15,000 milligram, and then after the that is done, that then affinity, after affinity chromatography, the end result is your 1.75, right? That is 35% is the yield, and the purification level is around 300,000 as compared to another chromatography. So highly recommend is the affinity chromatography, which is done with the primary and secondary antibody and seems to be the best one. Now once you have done, you have done the isolation, you have done the purification, now it's time to check that which proteins, to confirm that of which amino acids your protein is made up of. What is the sequence of amino acids of your protein? What is the sequence? Yeah, so in order to identify that, you do the Mardi-Toff spectrometry, in which you have a protein sample, you put over the matrix, yeah, and then you put laser beam on it. So laser beam go out of the beam splitter, and then via the prism it's get like very dense, and then hit over the matrix. Then it will ionize our sample, will make them into the positive charge, and they will go through this streaks of the ion source, and they will make into the small molecules into this flight tube, and the lightest ions will arrive at the detector first. So these ions we can detect and transfer them into the amino acids sequence. So Mardi-Toff basically means metric assisted laser desorption ionization, that is time of flight. So a laser beam ionized proteins embedded in matrix, ionized proteins accelerated towards the detector, and the detector monitors the arrival of ions as the laser triggers the clock. So in the end we get these kind of peaks. This is the intensity and this is mass to charge ratio. So there is insulin giving a value of 5733, whereas the beta galactoglobin giving a value of 18,364. Yeah, so both giving a different values here. So some different peaks. So now let's check how Mardi-Toff, Toff is done in the lab.
Rooker Maldi-Toff process. Maldi-Toff analysis provides additional structural information for target molecules. As Maldi-Toff is a two-phase process. Fragmentation can occur by application of higher laser energies, known as LID, laser-induced dissociation, or a background gas can be introduced to promote ion collisions. This process is known as collision-induced dissociation, or CID. Samples are co-crystallized with a suitable matrix on a sample carrier. The samples are vaporized by the laser into a vacuum while being ionized at the same time. During this process, the sample molecules can become excited. The now charged and excited particles are accelerated through the application of a high voltage, and fragmentation can occur as they travel through the flight tube. In this process, precursor ions and their respective fragments travel together at the same speed. Based on the expected time of flight of the precursor ions, specific ion packages may be individually selected, and all other ions blocked from continuing through the flight tube. The selected ions are then re-accelerated, with the precursor and fragment ions traveling at different speeds towards the reflector detector. The ions travel via the reflector towards the detector, and thus a top-top spectrum is recorded. The detection of the selected precursor ion and its fragments creates a top-top spectrum. Whether fragmentation occurs through the use of higher laser energies or a collision gas, the structural details derived from the MALDI top-top spectrum provide a deeper analytical insight of the targeted compound.
Yes. So that was the basics of the MALDI. Now one application of these MALDI top in order to find out the good results at the end, at how I will share all these videos later, students.
Today, we rely heavily on biotechnology for many of the necessities of life. For the food we eat, the clothes we wear, fuels for transportation, earth-friendly plastics, pharmaceuticals to treat our illnesses, and dietary supplements to help us stay healthy. Many of these products are produced using three core biotechnology processes: fermentation, recovery, and purification. Fermentation is basically cell farming. We program cells to produce a product. We nurture them as they grow and reproduce, and then we harvest them. In recovery, we separate our product from the cells where they were housed. And then in purification, we go a step further by removing everything else that's contaminating our product, leaving us with a very pure, concentrated solution. In this program, we're going to look at a typical purification process used in the manufacture of GFP, green fluorescent protein. GFP is broadly used as a biological marker because it's very well tolerated by most cells and doesn't interfere with normal cellular function. We'll examine the technologies, equipment, and materials used, how to prepare for the process, and how the GFP purification process is managed, step by step. There are two main operations used within purification: chromatography and filtration. Before we can appreciate exactly what these process steps are accomplishing, we need to take a closer look at what our green fluorescent protein has been through so far. Back in recovery, our harvested cells from fermentation were homogenized. Our target protein was inside each cell, so to get at it, the cells had to be ruptured. Homogenization freed a flood of cellular components, including membrane debris, cytoplasm, DNA, and proteins, including our target protein, GFP, and they were all mixed into the buffering solution used to suspend the cells. Most of these solids were then removed through centrifugation, or by centrifuge, but the liquid that remained, the clarified lysate, was still rich in biological products, including dissolved chemicals, proteins, and other impurities. Amazingly, through purification, we can target a single protein within this biological soup. In our GFP purification process, we'll be using multiple types of column chromatography. The column is a cylinder filled with glass, ceramic, or polymeric beads, which are engineered to interact with or bind with molecules based on one or more physical properties. Chromatography relies on differences. Each molecule has a unique set of physical characteristics, such as size, charge, or extent of interaction with water. Chromatography uses these differences to separate the target protein from other proteins and chemicals. Sometimes size is used to differentiate. Some beads have small holes in them and can temporarily trap or at least slow down smaller molecules as they travel through the column of resin beads, while molecules too large to enter the pores move around the beads and exit the column first. This type of chromatography is called size exclusion. In the case of charge, opposites attract, so a negatively charged chromatography bead will attract and bind to positively charged components in the process stream. Likewise, a positively charged bead will bind negatively charged components in the process stream. This charge-based chromatography is called ion exchange chromatography. And then there's water. Molecules that readily interact with and dissolve in water are called hydrophilic, water-loving, while those that don't are called hydrophobic, water-hating. Proteins contain regions that are hydrophobic and regions that are hydrophilic. Because water tends to form a shield around the hydrophobic patches within the proteins, they are not exposed to interact with the resin beads. By adding salt to the protein solution, we remove the water shield, exposing these hydrophobic patches on the protein and resin so they can interact. This is how HIC, or hydrophobic interaction chromatography, works. In our process, which uses ion exchange and hydrophobic interaction chromatography, the chromatography equipment is housed on a skid to make it compact and mobile. The main part of the apparatus is a glass column filled with resin beads, but we also have pumps to move the clarified lysate through the process, a supply hose and port to feed the column, a pre-filter to remove any remaining particulates, usually solid cell debris that has not previously been removed, before the clarified lysate enters the column, an exit port for the processed solution, and auto switching valves for directing processed solution to either waste or collection. To help monitor the chromatography equipment and the solutions flowing through the unit during the process, a number of sensors are located along the product flow path. There is an electrical conductivity sensor at the column inlet, a pressure sensor just before the pre-filter to help identify a filter clog, a flow meter to measure the rate of solution movement through the column, and an air sensor to ensure that no air has entered the flow path. As a solution leaves the column, it passes a UV sensor that reads optical density, a second conductivity sensor, and a pH sensor that measures how acidic or basic the solution is. The conductivity sensors let us know when a new buffered solution has filled the column. When the conductivity reading on the exit of the column matches the reading from the sensor at the inlet of the column, then we know the new solution has completely displaced the old one. The ultraviolet UV sensor monitors the concentration of protein in the product by observing the optical density of the passing solution. This sensor works hand in hand with the valves on the exit of the column. Through the controller program, we can set a protein concentration threshold. When the optical density of the solution leaving the column is below the threshold, the valve directs the flow to waste. When the optical density of the solution leaving the column is at or above the threshold, this means that solution contains our purified product and the solution is directed to a collection vessel. TFF, tangential flow filtration, is at its heart a simple process step. we're going to pump a fluid through and across a special type of filter known as an ultrafiltration membrane. The size of the pores in the filter material determines what passes through and what's held back. As with almost any filtering process, we can choose what we want to keep. The solution that passes through the membrane is referred to as the permeate. Because the pores of the ultrafiltration membrane are small enough to keep the product from passing through, the permeate contains no product and is sent to waste. The portion of the feed stream that does not permeate the membrane is called the retentate. It contains the retained product and is the stream we are most interested in. What makes TFF different is a core technology that enables it to be faster, more efficient, more flexible, and even self-cleaning. In conventional or terminal filtration, a fluid is pumped directly into a filter. The particles within the stream that can't fit through the pores of the filter build up at the filter surface, eventually clogging it. In tangential flow filtration, the stream moves across the filter, that is, tangential to the filter, instead of directly at it. The cross-flow current actually picks material back out of the filter media or membrane and into the stream. This retained material, called retentate, is recirculated to the supply tank and will continue to loop through the filter for as long as the process runs. We'll be using TFF for two different tasks within the purification process: concentration and diafiltration. We are processing purified GFP from a chromatography step. The fluid from the chromatography step is purified green fluorescent protein dissolved in a buffering solution. In diafiltration, we add new buffer to the GFP retentate while displacing the old buffer, effectively exchanging buffer solutions. Note, the GFP is retained by the membrane. If we don't add a new buffer, then we're concentrating our solution. Concentration is simply the removal of water and buffer components from the feed solution that results in a more concentrated solution of green fluorescent protein. From operation to operation, or product to product, the purification process can look quite different. It could be as simple as a single chromatography step, one cycle of TFF to concentrate the product, and final filtration. Or it could involve several different types of chromatography, diafiltration between chromatography steps, and a final conventional filtration. Our green fluorescent protein process is pretty typical of the process for biopharmaceutical products. The clarified lysate will be pre-filtered, go through anion exchange chromatography, and the GFP collected and pooled. Next, ammonium sulfate will be added to make the solution high salt, followed by hydrophobic interaction chromatography, HIC. The GFP is then collected and pooled again. The solution then goes through a TFF ultrafiltration step, which includes concentration of the GFP followed by diafiltration to exchange buffer solutions and remove salt. And the process finishes up with final filtration into bulk bottles or bags. It's time to gather everything we'll need for the purification process and ensure that the area and equipment are ready to go. The chromatography skid and TFF system are checked for proper operation. Process hoses are attached and examined for leaks. We verify that we are using the correct column resin and that the resin is properly packed. The column is filled with a storage buffer, and the product path is checked for trapped air and purged if necessary. Our raw materials include the clarified lysate from the recovery process, various buffer solutions that are tailored to specific process steps, and ammonium sulfate that we add to one of the buffers to make it high salt. The purification process is managed through the use of a batch record. There is a separate batch record for each processing operation. These documents lead the operator through the process step by step. with each step requiring a sign-off and separate verification by a second operator. The batch record also includes spaces for documenting times, activities, operation steps, and instrument readings. Before the process can begin, the purification area must be cleaned, disinfected, and organized. Any unnecessary equipment or materials are removed. All equipment must be cleaned, sanitized, and set up as required by standard operating procedures. All required materials and documentation must be gathered and prepared before the process may begin. The purification process begins as the transfer tank of clarified lysate from the recovery process is connected to the inlet pump on the chromatography skid. The first chromatography step in our green fluorescent protein purification process is anion exchange. At this point in the process, the pH of the clarified lysate is about 8.0, which means that the protein is negatively charged. Because it is negatively charged, GFP will bind to the positively charged anion exchange resin. The pump draws the lysate from the vessel, past the first conductivity sensor and pressure sensor, and through the 0.45 micron pre-filter. The pre-filter removes any residual cell debris or other particulates that may have contaminated the solution. If the pre-filter begins to clog, the pressure sensor at the inlet side of the filter will register a rising pressure, and the controller will signal the need for a filter change. After pre-filtering and before the column, the lysate passes through a flow meter and an air sensor. Then, as the lysate passes over the resin beads, the negatively charged protein binds to the positively charged beads. The solution leaving the column passes a UV optical density sensor, a conductivity sensor, and a pH sensor. The optical density sensor's low readings confirm that the GFP is not in the solution, so the outlet valve sends the solution to waste. When all the lysate has entered the column, or when the capacity of the beads to bind the protein has been reached, it's time for elution. Elution is the release of, in this case, green fluorescent protein from the beads by using a new solution, in this case a buffer that includes NaCl, sodium chloride solution. As the new buffer is pumped through the beads, at some point the GFP no longer binds to the beads and is released into the buffer. The resulting product stream is usually referred to as the eluate. The UV optical density sensor, which measures protein concentration, indicates when product begins eluting from the column. At this point, outlet valves are switched to allow flow of the eluate, the product stream, to a collection vessel. When the UV sensor indicates that all of the GFP has come off of the chromatography resin, the outlet valves are switched to waste. When all of the eluate has been collected and pooled, the anion exchange chromatography step is finished, and it's time for HIC, hydrophobic interaction chromatography. Hydrophobic interaction chromatography is based on the principle that hydrophobic chemicals on the resin surface will bind to hydrophobic patches on the GFP protein. In order for this to occur, the resin and protein eluate have to be in a high salt environment to remove the water shielding. The salt we use is ammonium sulfate. To remove the attached GFP protein from the HIC column, we simply lower the salt concentration during elution, causing the water shielding to reform, and the GFP protein detaches from the resin into the elution stream. The protein-rich eluate is collected and pooled. The product is now ready for the last major step, TFF. At this point in the process, tangential flow filtration will be used to concentrate and diafilter the GFP product stream. The eluate is rich in green fluorescent protein, but it's still too dilute and too high in salt. As the solution moves through the TFF apparatus, it leaves the supply tank, is pulled through a pump, past a pressure sensor, and then across the filter membrane. Everything that passes through the membrane, including the buffer solution, is known as permeate and, for this process, is sent to waste. The GFP protein is larger than the pores of the membrane and therefore is retained. The retained material, called retentate, is recirculated to the supply tank. Recirculation of the feed continues until the desired concentration of GFP is achieved. Following concentration, and while the protein solution is recirculating, a new solution, a storage buffer, is added to the feed. In effect, the protein is being washed by the flow of a new buffer solution in and the old buffer solution out. As this diafiltration step proceeds, the buffer solution that is being added to the feed replaces the buffer solution that the GFP was originally in, effectively removing any remaining salt as well. When this process is complete, the GFP solution is routed through a 0.22 micron final filter and then collected in appropriate containers, usually bottles or bags. The purification process is complete. The green fluorescent protein concentrate can now move downstream to final fill finish to be freeze-dried and packaged.
Okay students, that's it for today. I will share all these things later with you. I think, but if you watch, I have shared this drive with you. You can have every video in it actually. So just check in it, you will get everything then. Okay, thank you very much. Wish you all nice day. Yeah, take care. Bye bye. See you. Oh, it's going on.